Hello. So today I'm going to just be discussing chapter 2 from the microbiology book. So this chapter, it explains the properties of light and how microscopes work and the different types of microscopes used in microbiology. And some common staining concepts, techniques that identify the microorganisms. So understanding these concepts is important because microorganisms, they're too small to see with the naked eye. So scientists rely on microscopy to study and diagnose and identify infectious organisms. Okay. Now, light behaves as a wave. Two important properties of light are wavelength and the frequency. The wavelength is just the distance between two peaks of a wave, right here and here. And the frequency is the number of waves that pass a point during a specific amount of time. So how many waves? So the shorter the wavelength, the higher the frequency. So these two properties, they're related. The longer wavelengths have the lower frequencies, and the shorter wavelengths have the higher frequencies. That relationship is important because wavelength directly affects how much detail that a microscope can reveal. So here you can see the wavelength is measured from one peak here to this peak on the waves, and the amplitude is measured from the center line from the top of the wave to the middle right here. Okay. And light can be reflected and absorbed and transmitted when it encounters an object. So the difference, absorption is when the energy from the light is taken in by the material. So it's absorbed, like it devours that. And then transmission is when it passes through. That's why transparent items are hard to see, because the light is transmitted through that object. And then absorption, the transmission, the reflection, light bounces back. Think of like a basketball or some kind of ball. You bounce it against a wall, it's reflected back. And then one of the most important um, concepts of microscopy is refraction. Refraction. That's when the light changes direction. Um, Refraction occurs like when it passes from one medium to another, like from air into glass. So the bending of the light allows the microscope lenses to focus on the images, such as right here. Notice how the convex lens here. Now what it does, it bends the light to a focal point right there. And that's the point, that's what a microscope does. It bends that light to pinpoint a certain point. So it allows it to focus more. And then the visible light, it's only one part it's only a small portion of the electromagnetic spectrum. And the spectrum, it ranges from high energy gamma rays to low energy radio waves. So here, the visible light falls between the ultraviolet light and the infrared light. So this would be your visible. Now, while the red light has the longest wavelength, right here, you see it has the 700. It has the lowest frequency. Right here, it has a 10.6. Microscopes just use different portions of the electromagnetic spectrum depending on the type of microscope being used. Now, notice how the wavelengths increase like from the left to the right, and then the frequency decreases from left to the right. All right, and then there's 3 important terms that they use in microscopy, and that's the magnification, right here, and resolution, and contrast. So magnification just refers to how much larger the image appears, compared to the actual object. So it's how big it looks. And the resolution is how clear. Like it distinguishes between 2 separate points as separate objects. So it's able to separate it clearly. And then the contrast refers to the differences in the brightness that allows the structures to be distinguished from each other. And a microscope may have high magnification, resolution is poor, and the image will still appear blurry. But the best microscopes have all 3. They're enlarged, sharp, and visible. Now, early microscopes, they began a whole new world. There are actually many scientists that help contribute to the development of the microscope. Like Antony van Leeuwenhoek. Now they called him the father of microbiology because he was the first to observe a microorganism, which he called those animalcules. And Robert Hooke used a compound microscope to examine a cork. There. And he was the first one to actually describe cells. And Galileo, which is the more common name everyone knows, also contributed to the development of the early microscopes. Now, their discovery was actually so important because without the discovery of the microscope, you wouldn't be able to see the microorganisms and disease and bacteria would just be out of control. All right, and then the bright field microscopy, the lab standard, that's your average, everyday, what you see now in labs. It's the most common. It uses visible light and produces a dark image on a bright background. The total magnification is very neat. So, for example, a 10 times ocular lens, and your ocular lenses are here, your eye pieces. So, a 10 times ocular lens and a 40 times objective lens, which are these that you can change and alternate, would actually equal 400 times magnification of that. Excuse me. Now, obviously, oil immersion does improve the resolution and the stain, improve the contrast, make it stand out more. But here you'll see this is where you hold the specimen, and then the diaphragm is here and condenser, and this is where the light shines up, and here you adjust the light sensitivity and the focuses. So just remember your ocular and times your objective is your total magnification. All right. All right, so fluorescence microscopy uses fluorescent dyes called fluorochromes. So these dyes absorb energy and emit visible light, causing specific structures to glow. So kind of like fluorescent colors, they glow. So the fluorescence microscopy is commonly used to identify like pathogens and cellular structures. Like the bright cells stand out extraordinarily on a dark background. The confocal microscopy uses lasers to scan multiple layers of specimen and create highly detailed three-dimensional images. All right. And then the confocal microscopes are especially useful for like examining thick specimens like biofilms. Like they use these, like that's the 3D cell, that's the all right, and then here, the perfect example of the fluorochromes. They glow pretty colors, I think. All right, and then see, let's go on. All right, so most microorganisms are nearly transparent. So that transmission, like I said, goes through most. So the staining is used to make those more visible and add contrast to be seen. And simple stains use a single dye. Now, differential stains use, here we are. All right, the differential stains use multiple dyes to distinguish organisms or structures. So the examples are like the gram staining and endospore staining and the flagell staining. Right. So gram staining is one of the most important staining techniques in microbiology. The process includes 4 major steps. The crystal violet, it's the primary stain added to specimen smear, and it usually stains the cells purple or blue. And and
What are you doing?
Huh? The thick followed. So I'm sorry. The gram-negative bacteria lose the crystal violet during decolorization and appear pink after the counterstain. Right here. As we move through the steps, just notice the gram-positive cells stay purple. They stay purple. They hold that dye. And the gram-negative fade more to pink. All right, and then the other differential stains are just the acid fast, the endospore, flagella, the capsule. Okay, so during our clinical focus, during the chapter, Cindy, she developed a wound infection after scraping her knee. So the dark field microscope showed spherical bacteria arranged in clusters. And a gram stain revealed purple, gray-black clusters indicating a gram-positive cocci. So further testing identified the organism as a Staphylococcus aureus, a common skin infection. In this case, it just demonstrates how microscopy and staining techniques help healthcare providers pinpoint the pathogens and make sure they select the appropriate treatment for that pathogen. So in all, microscopy is an essential tool in microbiology. I mean, understanding light, lenses, magnification, resolution, staining techniques, it's allowed scientists and healthcare professionals to pinpoint the microorganisms and diagnose diseases, and treat those. And they continue to improve in their ability to study organisms that can't be seen with the eye. So thank you for listening.